Okay. Good. Okay.
Also, is your screen still sharing in Zoom? Yeah, you're gonna, yeah, sure. Yeah, thank you.
Okay. From the same cell. I'm getting a bit of feedback now after.
Now there's feedback on the computer.
Yeah.
Just 1 second.
Okay. Sorry, guys. Still feedback.
You're good.
Okay. All right. So we use these assays to map regulatory information in individual cells, and using this information, we can define properties of individual cell types. And so I'm going to gloss over the details of how we go from the profiles generated from individual cells to cell type specific regulatory programs, but it involves a series of processing steps and clustering and defining. Okay, sure. All right. Okay. Okay. Anyway, so we can define regulatory programs of individual cell types. Okay. So we've applied these techniques previously in several papers, one of which Todd mentioned, which was published a couple of years ago, where we profiled gene expression and accessible chromatin in individual cells in pancreas tissue as well as peripheral blood. And I'm showing you here on the left, this is the clustering of the attack-seq profiles, which has cell types in pancreas and other immune cell types in the peripheral blood, as well as matched RNA-seq profiles from the same cell types. And using this information, we can define the regulatory programs of individual cell types. So on the right, this is cis-regulatory elements that are active in each cell type in pancreas and peripheral blood. And so that tells us about which regions of the genome are active and which cell types, and we can use that information to understand how genes are regulated in each cell type. Another way that we use this information is to overlay this with genetic risk of type 1 diabetes. And in doing so, the combination of the location of regulatory elements in specific cell types and variants that affect risk of type 1 diabetes, the combination of those two things can tell us something about not only processes that are affecting regulation of these cell types, but perhaps processes that are causally involved in disease. And this is particularly relevant because most of the risk variants for type 1 diabetes are non-coding, don't affect protein coding sequence, but map to non-coding sequence and likely affect gene regulatory activity. So there's many risk variants for type 1 diabetes, many of which will overlap regulatory elements active in specific cell types. And by understanding how these variants function, which cell types they overlap and affect, which genes are being affected by these elements, and so forth, we can understand causal processes that are involved in disease, as well as the direction of effect, right? So we can get some insight into does increased or decreased activity of a regulatory element or gene increase or decrease risk of type 1 diabetes. So it's really the combination of cis-regulatory maps and gene regulatory information, specific cell types, and genetic risk information for type 1 diabetes, and the combination of the two that is our lab's primary focus. So again, this was something we did in the study that was published a couple of years ago, where we performed a GWAS study. So we identified genetic variants affecting risk of type 1 diabetes in hundreds of thousands of individuals. And so we have variants spaced across the entire genome that affect disease risk, most of which are non-coding again. And then we combine this on the bottom left with genomic maps in pancreas and peripheral blood, and the intersection of the two tells us something about which of these regulatory elements and cell types is potentially contributing to disease. So on the right, one example of this, just to exemplify how this works. So there are risk variants for type 1 diabetes that map near the CFTR gene. So CFTR is the cause of cystic fibrosis, which is also a major cause of diabetes, cystic fibrosis related diabetes, but had previously not been implicated in the development, as far as I'm aware, in type 1 diabetes. But these risk variants map in a non-coding region upstream of CFTR in regulatory elements that are active specifically in pancreatic ductal cells. And this regulatory element is regulating the CFTR gene. We did a further work to establish the direction of effect. So in essence, reduced activity of this regulatory element in ductal cells leads to reduced activity of CFTR in ductal cells, which increases risk of type 1 diabetes. So we can combine these genetic data with these genomic data to try to get insight into causal processes of disease. So I'm going to be telling you potentially about 4 things today, time pending, about how we're building on these studies to try to understand type 1 diabetes risk in the pancreas. The first study I'll talk about is mapping changes in regulatory programs in pancreas cell types in type 1 diabetic individuals and people who are positive for type 1 diabetes autoantibodies. And this is also a project that we've worked closely with Mark and Arena to select samples from NPOD to profile for these studies. And also just to note, I'll just say this once. So I've listed out here mostly graduate students, some postdocs, most grad students in our lab working on these projects who have obviously done all the work and contributed substantially to the success of these projects. Okay, so this first project is building on the previous study, which was essentially just mapping regulatory information in individual pancreas cell types and immune cell types in the baseline state. So next, we were interested in understanding how these regulatory programs change in type 1 diabetes and like pre-disease states, autoantibody positive individuals. And so for this project, we have pancreas tissue from NPOD for 12 control samples, 9 individuals who are positive for autoantibodies, and then 12 type 1 diabetics, about half of which had recent onset type 1 diabetes, and the other half had a longer term duration of type 1 diabetes. And for all these samples, we performed the assays I mentioned before, single nuclear RNA-seq to measure gene expression, ATAC-seq to measure chromatin and regulatory elements, and then in a subset of samples, this paired ATAC-seq and RNA from the same cell, from only a small number of these samples. And so what we're using these data to ask is, can we find changes in pancreatic cell types in terms of their abundance, in terms of the cell signaling relationships between these cell types, activity of regulatory elements, in many cases are accepting those signals, and then finally changes in gene expression level. And so I'll show you some examples of how we've been able to chart changes in disease, mostly in beta cells, but there's other cell types that we have some insights into as well. So this is showing you first the clustering of all the RNA-seq data that we generated from these samples. So this is 33 NPOT samples in total, whole pancreas, frozen tissue. From these samples, we identified 18 cell types and in some cases subtypes of cell types from about 275,000 cells. The way that we annotate the identity of cell types, for those not familiar, after clustering, is that you look at the expression of known marker genes for specific cell types. So for example, this cluster here expressed CFTR, so that's how we know they're ductal cells. This cluster down here expressed insulin, so that's how we know they're beta cells and so forth. And so we have annotation of each of these cell types, and I'll talk about in a minute, some insight into potential subtypes within certain cell types. On the bottom right, this is showing you, since we're generating data from a large number of samples, integrating all these data to define cell types is a major aspect of the data analysis. So this is showing you different characteristics of the data to demonstrate that it's well integrated across different donors, different technologies, because we have some RNA-seq, some multi-omic data, and then also different disease states. And so, you know, that demonstrates that there's not massive heterogeneity in terms of any of these variables in terms of defining the cell types in these data. So that's the RNA map, right? So that gives us gene expression profiles in each cell type. The next thing is ATAC-seq, so that's accessible chromatin profiles in each cell type. So again, we have 33 samples where we perform clustering. To identify the identity of the cell types from ATAC-seq, we use a technique called label transfer, which basically learns the identity of the ATAC-seq cells from the RNA-seq profiles. And we estimate that process, which is computational transferring of labels from one modality to the other, is about 90%, 8% accurate in terms of this transfer. So we're fairly confident that this is accurate. And the reason that we know this is because we have some multi-omic data where we treated it as either RNA-seq only or ATAC-seq only. So we pretended like we didn't know it was from the same cells. We know the true identity of the cells in ATAC-seq, and the estimated accuracy is through that comparison. So here we find a bit fewer cell types, 14 cell types from 152,000 cells. So from this information that we derive from accessible chromatin, we can map cis regulatory elements in each cell type, which is shown here. And then we also generate predictions of genes that are being regulated by each of these cis regulatory elements, which for those of you who study genomics is a major challenge, given that many regulatory elements are far away from the genes they're regulating. It's not always immediately obvious which gene is regulated by a cis regulatory element. And so these predictions that we generate give us some insight into the genes likely targeted by these cis regulatory elements. In many cases, can be quite far away from the genes that they're regulating. Okay. So as I meant, so that's maps from NPOT samples of both RNA and ATAC-seq. So one thing that we noticed in these data was that there were multiple subtypes or subclusters of exocrine cells, particularly for acinar cells, but also for ductal cells. For ductal cells, these have been characterized previously. So there's a Mus5b positive subcluster of ductal cells that's been reported before. For acinar cells, there have been some reports of heterogeneity in acinar cells, but it's a bit less clear exactly what that heterogeneity represents. So in our data, we identified 4 different subclusters of acinar cells. And if you look at the genes that preferentially are expressed in those different clusters, we identify processes that suggest that these subclusters may represent either cell states or distinct subtypes of acinar cells. So for example, there's a high enzyme cluster of acinar cells, which preferentially expresses digestive enzymes and so forth. Another subpopulation of SNR cells preferentially expressing cytokine signaling genes and so forth. So we think that's maybe a stress response subcluster of SNR cells and so forth. And in addition, if we look at the chromatin profiles of these subtypes, we find motifs for certain transcription factors that are preferentially active that again suggest potential drivers of this heterogeneity. And this is the case for many cell types, right? And in beta cells, we previously reported heterogeneity. They are suggesting that there's insulin secreting and stress responsive beta cell states. And I think for many cell types that this is the same phenomenon where there'll be multiple different cell states of these cells. And since in whole pancreas data, we're assaying a lot of acinar cells because the majority of the pancreas is acinar, we have increased power to, I think, identify heterogeneity in these cell types. So suggests that there are specific subpopulations of acinar cells within the pancreas. Okay, so we have our map of gene expression data, chromatin data, some insight into heterogeneity within certain cell types such as acinar and ductal cells. And so next we wanted to understand the relationship of these data to changes in type 1 diabetes. And so the first question we asked was whether the abundance of each of these cell types or subtypes changes from non-diabetic individuals to autoantibody to individuals who had recent onset or long-term type 1 diabetes. And as expected, we find a decrease in beta cell proportion, particularly in early type 1 diabetes. No evidence, as I was talking to somebody earlier, for changes in the abundance of beta cells in autoantibody positive individuals, as far as we can tell, but clear decreases in beta cell numbers in type 1 diabetes, as you expect. Also an increase in immune cell numbers in both autoantibody positive individuals as well as recent onset type 1 diabetes for macrophages, T cells, and particularly B cells, which are almost absent, at least as far as our ability to detect them in the non-diabetic individuals. So those are the major changes, right? And I think those are expected. Beta cells obviously decrease in T1D, although no evidence for change in autoantibody positive, and an increase in immune cell numbers in both autoantibody positive and early T1D. We also identified some evidence for changes in subtypes of acinar cells. So there was an increase in the high enzyme producing state of acinar cells in autoantibody positive individuals. This wasn't significant, but it was trending in that direction, suggesting that high enzyme population of cells may be increased in the stages preceding disease. Okay, so that's changes in abundance. So next we were interested in looking at changes in gene regulation and gene expression within each cell type. So this is showing you on the left here the number of genes that have significant changes in expression at each of the stages. So multiple autoantibody, recent onset type 1 diabetes, and long-term type 1 diabetes. So a couple things to note here. So first off, we see the largest changes in beta cells in early and late T1D. Other cell types, such as acinar cells and some other cell types, have some evidence for changes specifically in the recent onset T1D. But what is also quite noteworthy is there's no genes that have significant changes in multiple autoantibody positive individuals. Doesn't mean that there aren't any changes in these individuals, right? So we have like a limited number of samples we're working with, but it probably does mean that there aren't dramatic changes in the regulation of any of these cell types, at least those that we're able to study here in multiple autoantibody positive individuals, particularly in beta cells, which runs counter to some other reports recently that have suggested dramatic changes even in single autoantibody positive individuals. So to me, that there are probably changes in these cell types at these pre-disease states, but they're probably subtle and not as dramatic as they may have been reported elsewhere. But we do find quite a number of changes in the early stages of T1D. And so this is what we're looking at here on the right, is the volcano plot showing you genes with up and down regulation in recent onset type 1 diabetes. This is using both NPOD data as well as data available from HPAP publicly. And so what we see is strongest upregulation of class 1 MHC genes, which I think is expected, although it's pretty prominent to see these as the most overexpressed genes, as well as some components of like MHC processing, in addition to genes that are cytokine responsive and stress responsive genes, such as STAT4 and so forth. So we see a very clear upregulation of genes involved in specific cytokine responsive and class 1 MHC processes in recent onset T1D. On the bottom right, this is showing pathways that are broadly enriched for these upregulated genes, which is kind of just saying what I've already said, that upregulated genes are enriched for antigen presentation, interferon, cytokine signaling type processes. So again, we're interested in understanding, and that's, I think, pretty well established, right? Like, I think people wouldn't probably argue too much with that those are the types of changes you observe in people who had recent onset type 1 diabetes, but of course, interested in the changes that are occurring before the onset of diabetes. And like I said in the previous slide, we didn't find much evidence of dramatic changes in beta cells or other cell types in autoantibody positive individuals. But what we did do is take the set of genes that were significantly upregulated in recent onset type 1 diabetes and ask what their effects were in the preceding stages in both multiple autoantibody positive individuals, as well as single autoantibody positive from using HPAP data. And what we find is that the concordance in the effects of these genes that are upregulated in recent onset type 1 diabetes is very high in the multiple autoantibody positive individuals, suggesting the same set of genes is changing in these individuals, but you can clearly see it's just to a lesser degree, right? So the effects of these genes are roughly half, of what you observe in recent onset type 1 diabetes. So this suggests that the beta cells collectively of these multiple autoantibody individuals is like some intermediate phenotype to what you're observing in recent onset type 1 diabetes. By comparison, in the single autoantibody positive individuals, there's much less concordance, right? And almost, I mean, there's a little bit of a relationship, but it's very subtle, suggesting that the changes at this stage are fairly weak. So to me, like, the conclusion here would be that multiple autoantibody positive individuals is at least not everybody, like Martha and I were talking about earlier, but collectively this may represent an intermediate phenotype state, like a pre-disease state, at least in the beta cells of these individuals, whereas single autoantibody positive individuals don't have much change. If we look at the processes on the right that are changing in recent onset type 1 diabetes and multiple autoantibody positive individuals, so we see some processes that are upregulated in both, so antigen processing, presentation, cytokine signaling, interferon signaling, so forth. Some processes that are downregulated in both, so regulation of beta cell function, some lipid metabolism for reasons I'm not quite clear on. We also see a subset of processes that are upregulated specifically in multiple autoantibody individuals and not in the type 1 diabetes individuals, which are processes related to mitochondrial function, mitophagy, autophagy as well as some cytokine signaling that's distinct from the cytokine signaling processes here on the right. So there are some similarities in the processes as well as some differences. But I think together highlights that multiple autoantibody positive individuals have some changes that are occurring in their beta cells. So this is to show you just specific genes that are changing to these stages. So class 1 MHC genes are like strongly upregulated in recent onset T1D, but some are also upregulated in earlier stages. STAT and IRF, so we see some family members of STAT and IRF that are upregulated. Notably, STAT4 is significantly upregulated in both early recent onset T1D as well as multiple autoantibody positive individuals, IRF1 and perhaps STAT1 as well. So there's certain family members within STAT and IRF that appear preferentially upregulated, not only in disease, but in the stages preceding disease. So that gives us some insight into processes that are changing in beta cells in disease, and particularly those that are preceding the onset of disease. We also looked at changes in chromatin that might be driving some of these gene expression changes. So there's certain transcription factor motifs we find that are up or down regulated in T1D compared to control that also have some evidence for having intermediate effects. So for example, IRF is upregulated in T1D, PDX1 downregulated in T1D, and in both cases it appears somewhat intermediate in the autoantibody positive individuals. And we also did some work on the right here to link these factors to gene networks that they're regulating that basically further confirmed that these types of factors, IRF and in particular, and other factors, or regulating genes that are broadly changing in type 1 diabetes. And these genes are mapping in pathways that are very similar to the pathways that I showed on the previous slide related to cytokine signaling and antigen processing. So this is suggesting transcriptional drivers of these processes that are changing in the progression to disease. Okay, so the last part of this part of the story is that we're interested in then, as I said, since we're use genetic data to try to understand causality, we wanted to know which of these pathways that's changing in type 1 diabetes may have some evidence for playing a causal role in the development of disease. And so to do that, we defined cis regulatory elements that are predicted to regulate genes in each of these pathways, and then asked whether these elements are preferentially harboring risk variants associated with type 1 diabetes. So on the top left here, this pathway would be enriched for type 1 diabetes risk because these elements are preferentially harboring disease risk variants. On the bottom left here, this would not be involved in type 1 diabetes risk because there's no risk variants for type 1 diabetes in these regions, right? So we can use the polygenicity of type 1 diabetes to ask whether collectively things tied to genes in certain pathways are harboring disease risk. And so this is what we find here on the right. So these are pathways that are enriched for harboring risk variants for type 1 diabetes. So again, we find interferon signaling, both interferon gamma and interferon alpha, beta, antigen processing, as well as lipid metabolism and also beta cell function. So each of these processes is linked to regions that are harboring variants associated with type 1 diabetes. Whereas some other processes that are changing in disease, so apoptosis, autophagy, mitochondrial function, so these processes are changing in the progression to disease, but have no evidence for harboring disease risk. So they may be consequences of disease as opposed to causal things that are causally involved in the development of disease, if that makes sense. Okay, so the final thing we did with these data was to look for changes in signaling networks, right? So we have changes within each cell type in terms of gene regulation, changes in chromatin. We next wanted to ask whether there are changes in the signaling relationships between different cell types in the pancreas. So to do this, we used ligand receptor analyses, where basically you infer relationships between cell types based on the expression of a ligand arising from one cell type and the receptor expression in a different cell type. And then you basically match up ligands and receptors from different cell types and then ask whether those relationships are changing in progression to disease. And so this is showing you at a high level changes that are both outgoing from specific cell types as well as incoming to specific cell types across disease states. So for example, beta cells have increased incoming signal in early T1D, SNR, and some other cell types as well, whereas outgoing signals are most prominent from immune cells and several other cell types, right? And if we then take beta cells and ask which cell types are specifically signaling to beta cells, we find evidence for a large number of cell types that have changes in their predicted signaling incoming to beta cells that changes in the progression to type 1 diabetes. And so some of these signals may be modulating beta cell function and contributing to the changes we observed in gene expression in beta cells. So this is showing on the left here some of these signals that are changing in both recent onset and autoantibody positive as well as long-term type 1 diabetes that are incoming to beta cells from other cell types, in some cases from beta cells themselves. So for the example, BMP5 that's produced by beta cells and then signals back to beta cells. Other molecules arise from immune cells or other pancreatic cell types. So granulin, another one I've highlighted here, is produced by macrophages and so forth. So these are candidate signals arising from certain cell types that may be modulating beta cell function in the development of T1D. And for several of these, we've taken these ligands and then we culture cells in vitro with these ligands to kind of see what effects that has on beta cells in a pronounced way. So in the case of granulin, when we culture a beta cell line with granulin, we find up and down regulation of many genes, and these genes map into processes that suggest that granulin may be protecting beta cells in some capacity. So you find down regulation of antigen processing, cytokine signaling, and so forth when cultured with granulin, suggesting that granulin may be a protected molecule in this setting. BMP5 is a molecule that pronounces beta cell function and can contribute to development and so forth. So maybe beta cells signaling to other beta cells to stimulate their function. So in both of these cases, these molecules that we're observing in actual diabetic or pre-diabetic individuals are regulating processes that may, in both cases, be somehow trying to protect beta cells from disease. Okay, so just to quickly detail some limitations of the study and then also next steps that we're working on now. So a couple issues I think that worth noting. First, the sample size of these studies is still pretty limiting, right? So we have 8, 9, 10 donors per group, which I think prohibits a deeper understanding of changes, particularly in autoantibody positive individuals, as well as I think of high interest specific subsets. So specific antibodies that people have, single or multiple. If you have multiple, which antibodies do you have, and so forth, as well as other subsets such as low or high genetic risk, which is another area of interest. And so what we're doing now is that we have a larger number of samples that Mark and Irina have sent us that are all non-diabetic from a range of autoantibody states, and we're using single cell assays to profile all these samples, and then with these data, hopefully doing deeper comparisons of these different combinations, both in terms of autoantibody status as well as risk score. I meant to fill that in, and I'm sorry that I don't have the numbers there, but we do have actually quite a large number of both high and low individuals that have high genetic risk within the non-disease state. So I think those will be valuable comparisons. The second limitation is that both the number and depth of single cell profiles per sample is pretty limited from these assays. You capture about 10,000 cells from the experiments, at least in theory. In reality, it's probably closer to 5,000 or 6,000. Most of the cell types we care about are rare in the pancreas, right? Immune cell types, beta cells, and so forth are amongst rare cell types in the pancreas. And so we're undersampling those cell types, particularly immune cell types, where we don't have much ability at the moment to define, subtypes of immune cells and so forth. And so to address this, we've started using combinatorial indexing based techniques. There's both assays that we've implemented in our lab, as well as kits sold by companies such as Parse that come out of technologies that people have developed, like ShareSeq, where you can generate up to 1 million profiles per sample. Also at greater depth in terms of the number of genes that you capture and the amount of information that you get per cell. So hopefully the increased profiling of these samples, which are quite precious, but have a lot of valuable information in them, I think deeper sampling of these samples in terms of cell numbers will also reveal more. Okay, so that's it for the first part of the talk, and may have to go quickly through the next parts. Actually, let me skip through this part because I think that's maybe least relevant to this audience, and then I'll go to just this part of the talk. So, so we have a lot of interest in not only changes in beta cells in type 1 diabetes, but other cell types as well. And in particular, our previous studies have implicated acinar cells and ductal cells as potential causally involved in the development of type 1 diabetes, which I think changes in these cell types have been observed, but I don't think it had been established whether these were actually causally involved in the development of the disease. And so our results suggest some causal involvement of these cell types in disease. And so we were interested in trying to further understand this. And so this is a project now where we're looking at proteins that are causally involved in type 1 diabetes risk using genetic data, single cell genomics data, as well as large scale proteomic data. So the kind of design of this study is our interest is to identify protein biomarkers of type 1 diabetes in exocrine cells, but other cell types as well. And to do this, we're using a technique called Mendelian randomization. Is anybody familiar with Mendelian randomization? No? Okay. So Mendelian randomization is it's kind of like a randomized controlled trial, but using genetic data. And the way that Mendelian randomization works, they're quite handy tools, is that you have a set of genetic variants that are associated with an exposure of interest. And that exposure, in this case, is the levels of a protein in circulating blood. Could be anything, though. Could be a phenotype, could be lipid levels, could be height, could be weight, could be whatever, right? And the key thing is that you have a set of genetic variants that's associated with that exposure. And then you ask whether collectively these variants are associated with some outcome, either increase or decrease risk of the outcome. And in this case, the outcome of interest, of course, is type 1 diabetes. There are some assumptions that we have to make here, and that these instruments are not associated with, say, confounding variables that may also influence your outcome. And so we have to do some work to make sure that that's not the case. But in essence, what Mendelian randomization is doing is taking a set of variants associated with some exposure and asking whether collectively they're associated with an outcome. And so what we're interested in here is asking whether circulating levels of specific proteins are affecting risk of type 1 diabetes. So our exposure is the variants associated with levels of each protein. Our outcome is type 1 diabetes risk. And to do that, we're using data that's generated using UK Biobank data, where they measured levels of proteins across many individuals. They also have genotype data for these individuals, and so they can map variants that affect protein levels. And then we're asking whether the variants associated with levels of each protein are causally associated with the development of type 1 diabetes. And that's using GWAS data that we previously generated. So I'm showing you here the results of these analyses, right? So for each of those 1,000-odd proteins, we asked whether the variants associated with that protein were also associated with type 1 diabetes risk using Mendelian randomization. And so what we find at the end is 16 proteins that have evidence for a significant effect on type 1 diabetes risk, some of which are protective and some of which are risk increasing, meaning that increasing levels of the protein on the left are protective for type 1 diabetes, and increasing levels of the protein on the right are increasing risk for type 1 diabetes. And so some of these may look familiar to you. There's some cytokine signaling molecules here, but then there's also some acinar proteins, digestive enzymes produced by acinar cells, such as PRSS2, which is trypsin, as well as some additional pancreatic enzymes, right? So that suggested to us that perhaps increased level of pancreatic enzymes, at least in circulating blood, is protective against the development of type 1 diabetes. And this is just to kind of reiterate that if we take those 16 proteins and then ask which pathways they map into 3 main pathways. The primary one is pancreatic, secretions or digestive enzymes, which is trypsin, and then CPA1 and CPB1. We also find enrichment for cytokine interactions. So these molecules are all involved in cytokine interactions somehow. And then also proteins involved in cholesterol metabolism. So again, each of these proteins, their levels, using Mendelian randomization, we can estimate that they play some causal role in the development of type 1 diabetes. And for the pancreatic enzymes, most of these are protective. So increased levels of the pancreatic enzymes show protection against the development of type 1 diabetes. And this is showing you the results here. So this is each of the variants that's associated with the levels of trypsin or these other pancreatic enzymes. And you can see pretty clearly that with increased variant, the alleles associated with increased levels of the protein are correlated with decreased risk of type 1 diabetes, right? And that's the lines are decreasing for all 3 of these proteins, right? So you can see that increasing levels of these proteins is again protective for type 1 diabetes. So that was interesting, right? And that kind of further supports our notion that pancreatic exocrine cells are playing a causal role in type 1 diabetes, although we don't really know how. But I think there's multiple lines of evidence now that support that exocrine cells are contributing to disease. And so one thing we did was to take these variants that were associated with pancreatic enzyme levels, which we know are collectively associated with protection from type 1 diabetes risk, and ask which cell types these variants preferentially overlapped with. I think we would presume that they're probably located in acinar cells because they're variants associated with levels of enzymes produced by acinar cells. And so we do find some enrichment for these variants in regulatory elements active in acinar cells, but we also find ductal cells as well, which may make sense because ductal cells are helping these enzymes pass through into the gut. And we also find evidence for cells in the gut themselves. So these are probably all cell types that are modulating the levels of pancreatic enzymes as they're produced by acinar cells, make their way through the pancreas into the gut, and then are further processed, okay? So this is supporting that this causal processes that these protein levels are contributing to disease is mediated through exocrine cell types in addition to other cell types outside of the pancreas, notably the gut. So yeah, so this is further evidence that exocrine cells are contributing causally to the development of type 1 diabetes. We can also use this information to understand the mechanisms of individual risk loci for type 1 diabetes. So for people that are interested in understanding how risk variants at specific loci operate, some of these risk variants are also strongly associated with levels of these pancreatic enzymes. So for example, this locus here, which has no known mechanism to my knowledge, these variants that are associated with type 1 diabetes are also strongly associated with trypsin levels in blood. So suggest that perhaps this locus is contributing to disease by modulating trypsin levels. And then if you look at the variants associated with type 1 diabetes here, it's kind of hard to see, but these map in regulatory elements active in S and R cells. And so therefore likely potentially modulating trypsin levels by affecting S and R regulation. So this gives us some insight into how this locus is functioning. So the final thing we did was to ask whether the levels of the proteins was changing in progression to type 1 diabetes. So this is a distinct project from the NPOD project that are moving at separate paces. So we didn't want to use the NPOD data for this project. So here we used data from HPAP, which are publicly available data in PancDB that we obtained from non-diabetic, type 1 diabetic, as well as type 1 diabetic autoantibody positive individuals. And this is mostly purified islets, so not whole pancreas. And so here, most of the cells are endocrine, although you do get some non-endocrine cells. And this is showing you the clustering of all these data from HPAP on the top left here. Again, we have good representation from endocrine cells, but then we also have non-endocrine cell types, acinar and ductal cells, and here are the marker genes for each of these cell types that show how we annotated them. So then we wanted to know whether the levels of these proteins were changing that we think have a causal role in type 1 diabetes, if they're changing in diabetic individuals and also in pre-disease states. So here are proteins that were causally linked to type 1 diabetes: trypsin, CPA1, CPB1, and each of these had some evidence for having a decrease in expression in type 1 diabetes. And then not so clear if there's a difference in autoantibody positive individuals, although at least for trypsin and maybe CPA1, that does seem like maybe there's a trend towards like autoantibody positive individuals being intermediate to non-diabetic and type 1 diabetic individuals. But I think this is further supporting that, it's showing that these genes are changing in progression of type 1 diabetes. And I think together with the evidence that increased levels of these proteins is associated with protection against type 1 diabetes, decreasing expression of these genes in people developing type 1 diabetes, I think may paint a consistent picture. But the mechanism of how these proteins are actually causally contributing to type 1 diabetes, I think is pretty wide open, right? So worth the smiling. So, but I think that's the big question, right? Is I think we have a lot of evidence that exocrine pancreas and things being produced by the exocrine pancreas are causally involved in disease. How that is pretty wide open, although I think is interesting to try to understand what mechanisms that may be. Could be things like direct signaling to beta cells, could be signaling through the gut back to beta cells, could be promoting inflammation. There's multiple mechanisms through which these proteins can potentially affect disease. And I think understanding that is obviously the important mechanistic question. Okay, so in the last couple minutes, I'll just talk about one final project that relates to creating resources for the community that people can use to ask questions of interest in terms of type 1 diabetes risk, as well as changes in the pancreas, and help to try to democratize the ability for anybody to ask questions like this for a gene or pathway of interest. So one attempt at doing this is recently on bioRxiv. It's in review now, where we took all the data from HPAP, single cell RNA-seq data, which they have 65 samples in HPAP at the moment. So HPAP is a great resource that makes all the data it generates available to anybody. The limitation of these data is that they just provide the raw data. So anybody who wants to make use of these data, it's not immediately clear, if you're not an expert in data processing or bioinformatics, like how this information is useful to you. So we took all these data and then clustered it and then tried to create resources that people could actually use these data and ask questions from these data and make it useful to the community. And the data are all at the site ilogenomics.org. So there's 65 samples in total, come from a range of disease states: type 1 diabetes, type 2 diabetes, pre-disease states. And in total, there's about 200,000 cells from these 65 samples. So what do we do with these data? So we have this clustering. We can define marker genes of specific cell types. We can define the expression of all genes across the genome in each cell type, changes in the expression of genes in disease. These maps can be used to integrate external data sets, so other single cell data, spatial data, as we were talking about earlier, visualize profiles, right? So look at genes of interest in various ways, as well as pipelines that we make available for anybody to use, right? So all the pipelines used to create all of these resources are free for anybody to take and use for whatever projects they're interested in. So I'm just showing you some examples here of how you can use the data. Again, going from raw data, provide an HPAP to things that are tangibly useful for people who are non-experts to glean insight from these data. So one thing that you can do is take a gene of interest and search for the expression of that gene across different cell types, or across different samples in HPAP per cell type. So for example, for MAFA, just a gene I picked, it's expressed in beta cells. So you can look at, in a tabular form, the expression of MAFA across different cell types, and it's expressed highly in beta cells and not in other cell types, or you can look at its expression across different samples. There's also visual tools that you can look at the expression of these genes. So if you look at MAFA expression across different cell types, you can see it's clearly very preferentially expressed in beta cells on the top here, as well as if you look at this on these UMAP plots, you can see its expression, same thing, is very restricted to beta cells. So you can search for any gene you want, right? And these are tools that enable you to look both in tabular format as well as visual formats, the expression of any gene you're interested in. The second thing we did was to create browsers for differential expression, right? So I talked a lot about differentially expressed genes and using NPOD data. So this is the same thing, but instead using HPAP data, where you can look for cell types of interest, disease states, type 1 diabetes, type 2 diabetes, autoantibody antibody positive individuals, and then look at the genes that have changes in expression in each of those states and given cell type, as well as search for specific genes of interest. If you want to know whether your favorite gene is differentially expressed in beta cells and type 1 diabetes, this allows you to look up that information. Yeah, one last thing you can do is use this as a reference. So this is a great reference of different cell types in the pancreas. And if you have your own single cell data that you want to map against this reference, you can do that. This is using a tool called Azimuth, which is just a computational method for mapping external data onto some reference. And so this is a reference we made from HPAP data. And it takes about like a minute to map your own data onto this reference. And that gives you automated identities of any cells that are in your data, right? So very, very fast way to map your own data to this reference to get cell type identity, and makes it easier to do things downstream. Also makes it easier to annotate what cell types your data are, which we work with a lot of different people who generate a ton of single cell data, and depending on their expertise or the type of data they're generating, and it may be quite laborious to try to figure out the identity of different cell types. So this kind of automates that whole process. It makes it a lot easier to do downstream work. And so finally, we make all these pipelines available. If anybody is bioinformatically inclined and wants to take these and use them for their own projects, different tissues or whatever, then these are freely available. Okay, so the last thing that we do in terms of providing resources is to create web portals to access genetic data for type 1 diabetes, right? So obviously, there's a lot of interest in understanding the genetic risk of type 1 diabetes and whether certain genes are involved in disease risk, or what the mechanisms are of how certain risk variants operate. But that can be a bit obtuse to people who aren't experts in this domain. And so these portals, the type 1 diabetes knowledge portal in particular, is a resource that lets you search for any region of the genome or any variant or any gene of interest, and shows you genetic data for those genes or region of interest. And you can ask questions, for example, about, does modulation of this gene increase or decrease risk? Is there evidence that variants associated with this gene are in regulatory elements active in certain cell types and so forth, and may help people gain mechanistic insight into how their favorite gene of interest is potentially operating. So those are resources. Okay, so to summarize, I'm not taking up too much time. So a total single cell genomics and other omics for certain projects can help reveal the role of pancreatic cell types in type 1 diabetes. So for the first project, using data generated from NPOD samples, right, broad changes in the abundance, regulation, signaling networks of pancreatic cell types in type 1 diabetes, particularly beta cells, but other cell types as well. And in addition to changes that we observe in multiple autoantibody positive individuals that kind of mirror those to a lesser degree than observed in recent onset type 1 diabetes, less evidence for changes in single autoantibody positive individuals, as well as some of these changes where we have some evidence that they're causally involved in driving risk of disease. Second project I didn't have time to talk about. This is more study looking at variants associated with regulatory element activity. And we find evidence for risk variants for type 1 diabetes affecting both beta cell as well as exocrine cell regulation. Third, which I did talk about, so circulating levels of pancreatic enzymes are causally linked to protection against type 1 diabetes. And this appears to be mediated through altered regulation of exocrine as well as other cell types involved in the chain of how these enzymes are moving through the body. And then last, we spent a lot of time trying to create web resources to host and visualize data derived from single cell data, genetic data for other people to be able to use them, so that hopefully people can take these resources and develop hypotheses about genes and pathways of interest in the role in type 1 diabetes. Okay, so I'll stop there. Thank you very much. And yeah, I'm happy to take any questions.
Oh, that was really exceptional. Do we have questions from the audience? If I can, let me just start. So when you say we have some time. So when you say causal, do you have an idea of when this is occurring in the exocrine pancreas? Are you this question sort of is chicken and egg, right? Is it that we're losing beta cells and islets and there's a proximity defect in tissue around it, or do you think this is truly causal all the way at the beginning and epigenetically regulated?
I think all we can say is that there's some genetic risk tied to things that are specifically affecting exocrine cells. So that would argue against it being secondary to an effect on beta cells, right? Like if the question is, does like loss of beta cells then some like insulinotropic effect on exocrine cells and you're losing like this. But so the variants affecting exocrine, in many cases, things that are specifically expressed in exocrine cells, right? Like CFTR, trypsin, right? They're not expressed anywhere else in the pancreas, right? As far as I know. So that these are very, very specific to exocrine cells. If the variants are affecting those genes, that would argue that the function of exocrine cells is causally driving some aspect of the disease process. I'm sure there's secondary effects as well, like beta cell effects then leading to changes in exocrine cells also, but that's probably not causal. So there's probably some combination of causal and just consequential effects. When it's happening, I think that's a great question, right? Like it could be early in development. Like this also link between pancreas mass we were talking about earlier and type 1 diabetes risk, right? So that's probably developmental, right? So there may be some developmental component of where this is acting, or it could be postnatally and like some aspect of fetal or adult exocrine function. But like where it's acting, I think is that's a good question, but we don't really know at the moment. Yep.
I think Melanie, you got a question.
Yeah, first I wanted to say I really like the type 1 diabetes knowledge, borderline knowledge.
Oh, cool. Excellent. Okay, great. Glad to hear it.
But my question is related to the increased pancreatic enzymes being protected for type 1. So I'm under the impression that for pancreatitis, that increased pancreatic enzymes are a risk factor for pancreatitis.
That's right. Yep. Yep. That's a very good question. I don't know. I mean, we have some actually evidence from our previous study that increased risk of pancreatitis was linked to increased risk of type 1 diabetes. So that would run counter to what we're showing here. It's possible that, so we're looking at levels of these proteins in the blood. So it's possible that's different than what is actually going on in the pancreas, and potentially, I don't want to say opposite, but there may be a complex relationship between what you're measuring in blood versus what is actually in the pancreas, right? So that's one possibility. Yeah, beyond that, I don't actually have a great answer. I mean, this is like we have this observation, and I think we're trying to understand it. When we do observe decreased expression of these genes and progression to type 1 diabetes, right? So to me, that also supports that that increased levels of them may confer some protection. But I agree, it's confusing given the relationship to pancreatitis. And like I said, there is a link between increased pancreatitis risk and increased type 1 diabetes risk. So in short, I don't know, but yeah, it's it's a good question, but I don't have the answer. And the best guess I have is that there may be some difference in what you're measuring in blood, like circulating blood, versus what's actually happening in the pancreas. Yep.
Yeah, just 2 comments, Carl. Fantastic presentation. I think I want to caution you, as Todd has said, associations or correlations do not imply causality at all. I don't see how you can make the conclusion that there's any causation with the defects in either genomics or proteomics. I clearly see that they're associated.
Yeah.
You know, with let's say multiple antibodies or new onset disease.
Right.
That was just one thing. The second thing was,
Let me address that real quick. So yeah, so changes that occur in disease processes may or may not be causally involved in the disease, right? Those are just changes that you're observing. The key is a link to genetics, right? Genetics is not changing, right? So, and these pieces, not all pieces, but some pieces that are changing in disease are also harboring disease risk. We haven't proven that they're causal, but the fact that they're harboring disease risk strongly suggests that they're probably on the causal, to me, pathway to disease, right?
It's just an association that if you have
Association between what, though?
you have a larger pancreas, more overall beta cell mass, and so you happen to have more trypsin in circulation, and that is associated with protection, but it's just because you have a starting larger mass of beta cells. It's true, but unrelated to the pathogenesis.
So just the
Yeah, I
argument
mean, yeah.
that The level of the enzymes probably normalized by something simple stuff, like normalized by the number of cells somehow, right?
Well, in the blood, it's just you're measuring levels in the blood, right? So, yeah, I mean, I get what you're saying in that context. Yeah, I mean, I don't think that, so like not all proteins in the exocrine pancreas are causally linked to disease, right? It's only a subset. So to me, so these variants are associated with, yeah, I mean, so in that case, I could see that being some mechanism that's not directly due to trypsin, but something that is related to the production of trypsin, could be the amount of cells that are producing it, right? But that's still, at the end, is linking back to something causally related to the exocrine cells that are producing those enzymes, right? But in that case, I get it. In the case of the changes in the cells that are harboring disease risk, that I find a harder time seeing how that could be correlative as opposed to causative, right? That there's processes where disease risk variants are lying. And so it's hard for me to see like where else that could be acting outside of the pathways that those variants are lying in, right? I get that we haven't proven, you know, if we like do like base editing or something of these variants and like do that level of proving, but I think the overall pattern suggests a causal link, not a correlative link, because why else would disease risk be present in those pathways if it wasn't causally involved in disease? For the enzyme levels, you could probably come up with reasons why that may not be directly causal itself, could be consequential. But for example, and we looked at, well, okay, so this is, maybe I didn't explain this very well. I mean, one of the big assumptions with Mendelian randomization is it's not, the variants of your exposure of interest are not associated with other things, confounders. So in this case, the variants associated with trypsin levels ideally are not associated with pancreatic volume, pancreatic size, things like that, right? And I think Ruth checked, and these variants associated with levels of these proteins did not appear to be like that dramatically associated with any other things that we could think of that could be confounding, including pancreas size. So we did look at that. Doesn't mean that that's not driving it, but you know, and it's hard to rule out all possible confounders, but ideally, if you're excluding all potential confounders, then all you're left with is the genetic effects and the exposure, and then you can draw a link between the exposure and the outcome. But I think practically, in many cases, it's hard to completely exclude all potential confounding. But we did look at the pancreatic volume. So these variants not strongly associated with pancreatic volume, but all strongly associated with trypsin levels, right? So that would argue against that it's mediated through pancreatic volume. Doesn't prove it, but you know, that's part of like the sensitivity analyses for these things.
You know, sort of along those lines, I was intrigued by your association with defects in lipid metabolism potentially, and there is some evidence that these may occur before the onset of antibodies. There is some evidence. But I was a little surprised too that you didn't see defects in perhaps glucose metabolism. That you know, either in genomic or proteomic analyses, because you didn't see this in multiple antibodies, but you did see it in new onset. In new onset, classical feature is diabetes, which is, you know, elevations in glucose. So I would have thought that you would have seen it either as cause or effect. You know,
I don't think we have, but we'll have to double check. But I'm fairly sure we didn't see that. But I don't want to say for sure, but
maybe.
I don't. Well, yeah, it wasn't that prominent, so it may still be there. But yeah, it wasn't very dramatic, I guess I'll say.
Any other questions? Like two or three in the chat.
Oh, yeah.
Pass through, they just then kind of continue. Scroll up again. Pass through a little bit. Move up a little bit. One more. There we go. First one.
Be thought to include controls from individuals with well-defined extract-specific diseases, pancreatitis, cystic fibrosis. So we do pancreatitis. I don't know where we'd get that from. Cystic fibrosis, we do have some pancreatic tissue from NPOT. This is a small number of CFRD cases, right, Arena, that we got with Rebecca in Washington, and then Rebecca also has another source for cystic fibrosis pancreas. So that we're looking at, although we don't have data at the moment. Pancreatitis, I'm not sure. But we don't have any data at the moment from pancreatitis donors. Okay, second question. Does it matter that the tissues were used were not from patients from type 1 diabetes and looking at the ATAC-seq? So that's a good question. So most risk variants for disease modulate the function of regulatory elements independent of those people actually have disease or not, right? These are risk variants, meaning that they're also present in many individuals who never develop disease, right? So they're just genetic factors that anybody has, many of which have very small effects on disease. There's plenty of people in this room, presuming, you know, almost none of us have type 1 diabetes. Our lab is a bit enriched as well, but most people don't have type 1 diabetes. All of us are carrying some of these risk factors, right? And they're modulating the activity of these regulatory elements. Most cases, regardless if you have disease or not, there's probably some interaction between like changes that are occurring in disease and the effects of risk variants, so that there are things like that that depend on disease states, but a lot of them just affect physiological activity of regulatory elements. That site's not working. I think we were looking at it earlier, weren't we, Martha? Yeah, okay, sorry. If it's, maybe try again, it should be up, but if not, email me and then I'll we can help you.
So if you were to speculate as to how, let's just take trypsin as an example. Do you think it's modifying the microbiome composition somehow because it changes the way dietary intake is modifying the gut?
It could. That's a good hypothesis. Yeah, it could be modulating immune cell function, right? I mean, I think enzymes can directly signal to immune cells as well, right?
Sort of cleave things off.
Yeah, or it could be cleaving proteins and then creating like antigens, right? So I think there's a lot of possibilities. And I guess all we can say at the moment is that it seems like these molecules have some evidence for being causally linked to disease, definitely are changing in the progression to disease, but how they're operating, yeah, I think it could be many things. Microbiome is definitely one option. Yeah. Do you see adipocytes in your data from whole pancreas? No, I thought that is what the first project is based on. Also, no. There's no, I don't know, maybe there's some adipocytes in pancreas. I don't, probably not normally. Somebody else probably knows this more than I do, but maybe it's small amounts. Oh, yeah, type 2, yeah. Or like, maybe cystic fibrosis patients, well.
All right, well, any other questions? Well, thank you very much. This is really.
Thank you for having me.